Hello my dear students, good morning. Welcome you all to a new video of Sunanda's tutorial. In this video I will discuss about the glyoxylate cycle. It is the part 5 video of lipid metabolism, and in this part I will discuss only about the glyoxylate cycle. What is glyoxylate cycle? It is the cycle through which fat is converted into the carbohydrate. This cycle is a variation of tricarboxylic acid cycle or TCA cycle or Krebs cycle, especially observed in plants and some microbes. Microbes like fungi and bacteria perform this glyoxylate cycle. Among higher plants, it mainly occurs during germination of oily seeds, where the stored lipids or oil are converted into sucrose, that means into a carbohydrate. Glyoxylate cycle does not occur in animals because they lack two specific enzymes which are required during the glyoxylate cycle, and that is isocitrate lyase and malate synthetase or synthase. These two enzymes are the key enzyme of glyoxylate cycle, and animals lack these two enzymes, so they do not perform the glyoxylate cycle. Then the location of glyoxylate cycle, it occurs in the glyoxysome, which is a special type of peroxisome present in germinating seeds. Glyoxylate cycle was first discovered by Hans A. Krebs and H. L. Kornberg in 1957. They jointly discovered this cycle. Hence, it is known as Krebs and Kornberg cycle. The particular role of this glyoxylate cycle is to convert two molecules of acetyl coenzyme A, which is produced from the beta oxidation or which is the result of beta oxidation, to succinate. So, the ultimate aim of this cycle is to convert acetyl coenzyme A to succinate, which is further metabolized. This succinate is further metabolized inside the mitochondria and cytosol by gluconeogenesis towards the formation of glucose and finally to sucrose. In glyoxylate cycle, the isocitrate is converted into the glyoxylate and succinate by isocitrate lyase instead of alpha ketoglutarate. That means in normal Krebs cycle, the isocitrate is converted into the alpha ketoglutarate. But in glyoxylate cycle, the isocitrate splits to form glyoxylate cycle and succinate. Let's discuss the glyoxylate cycle in details. So at first, after fatty acid oxidation, beta oxidation or alpha oxidation, acetyl coenzyme A is released. Then this acetyl coenzyme A reacts with oxaloacetic acid to form citric acid or citrate. Oxaloacetic acid or oxaloacetate is a 4-carbon compound. It reacts with acetyl coenzyme A, which is a 2-carbon compound. In presence of citrate synthase enzyme, it forms citrate or citric acid, which is a 6-carbon compound. Then citrate or citric acid in the presence of the enzyme aconitase gives rise to isocitrate. Then it is the key reaction of this glyoxylate cycle which make it different from the TCA cycle or Krebs cycle. Isocitrate, which is a 6-carbon compound, splits in the presence of the enzyme isocitrate lyase to form succinate, which is a 4-carbon compound, and glyoxylate, which is a 2-carbon compound. Then this glyoxylate again continues in glyoxylate cycle. Glyoxylate, but the succinate enter into the mitochondria where it is converted to malate. Malate again flows back to cytosol where it is converted into oxaloacetic acid, and oxaloacetic acid enter into the gluconeogenesis, and this glyoxylate again forms malate in the presence of malate synthetase and acetyl coenzyme A. This glyoxylate, by reacting with acetyl coenzyme A, forms malate, which is again a 4-carbon compound, because glyoxylate is a 2-carbon compound and acetyl coenzyme A is a 2-carbon compound, and finally they together produce the malate, which is a 4-carbon compound. Then malate, in the presence of malate dehydrogenase, converted into oxaloacetic acid. So at first fatty acid oxidation occur. From fatty acid oxidation, acetyl coenzyme A is released, which is a 2-carbon compound, which react with oxaloacetic acid to form citric acid or citrate in the presence of the enzymes citrate synthase. Then this citrate, in the presence of the enzyme aconitase, gives rise to isocitrate, which is a 6-carbon compound. Then this isocitrate split in the presence of isocitrate lyase to gives rise to succinate and glyoxylate. Succinate enter inside the mitochondria, where it is converted into malate. Then malate flows back to cytosol, where it is converted into oxaloacetic acid. Oxaloacetic acid finally goes or ultimately enters into the gluconeogenesis pathway. Then the glyoxylate, which is formed, converted into malate in the presence of acetyl coenzyme A and in the presence of the enzyme malate synthase. Then this malate again converted to oxaloacetate in the presence of the enzyme malate dehydrogenase. So it is all about the glyoxylate cycle and how it is different from Krebs cycle. Glyoxylate cycle is an anabolic process, but TCA cycle or Krebs cycle is a catabolic process. Glyoxylate cycle is anaerobic, whereas TCA cycle is aerobic one. So these are two main difference between glyoxylate cycle and Krebs cycle. So it is all about glyoxylate cycle, which is very important from examination point of view. I hope you really enjoyed this session. In my next video, I will discuss what is gluconeogenesis and what is its significance. So stay tuned and enjoy my videos like this. Thank you.